Hello, I'm Armin Hausa.
And my name is Babatunde Buhari.
And my name is Santana Burton, and today we're going to be talking about hyperinsulinemia effects on prostate cancer.
What is prostate cancer?
Prostate cancer is a disease in which cells in the prostate gland become abnormal and start to grow uncontrollably, forming tumours. Prostate cancer is a malignancy of one of the major male sex glands, along with the testicles and the seminal vesicles. The prostate secretes the fluid that makes up semen. The prostate is about the size of a walnut and lies just behind the urinary bladder. A tumor in the prostate interferes with proper control of the bladder and normal sexual functioning. Often, the first symptoms of prostate cancer is difficulty in urinating. However, because of a very common non-cancerous condition of the prostate, benign prostatic hyperplasia also causes the same problem. Difficulty in urination is not necessarily due to cancer.
What is a metabolic syndrome?
The metabolic syndrome refers to a cluster of cardiovascular disease, risk factors whose underlying pathophysiology may be related to insulin resistance. However, there is uncertainty as to whether all patients with the syndrome are indeed insulin resistant. So the etiology has been broadened to include concept of obesity, adipose tissue disorders, and other factors. The clustering of CVD risk factors with type 2 diabetes, hyperlipidemia, hypertension, and obesity was first noted in the 1960s to 1970s. The concept of the metabolic syndrome was proposed in 1988 when Raven published a landmark paper describing syndrome X. Subsequently, the name metabolic syndrome was coined to reflect the range of metabolic features involved.
What is hyperinsulinemia?
Hyperinsulinemia is a condition in which there are excess levels of circulating insulin in the blood, also known as prediabetes, insulin resistance, and syndrome X. If hyperinsulinemia is untreated, this can develop into diabetes or can lead to an increased risk of polycystic ovary syndrome, PCOS, hyperglycemia, or coronary artery disease.
Research suggested that energy balance and fat intake influence prostate cancer progression, but the effect of the dietary carbohydrate on prostate cancer has not been found yet. This research proves that hyperinsulinemia resulting from high intake of refined carbohydrates will cause more growth of tumors in the murine LNCAP xenograft model of prostate cancer. In 1987, when Raven discovered metabolic syndrome, skeletal muscle insulin resistance was being studied, and during these studies, they found out that high level of fat, refined sugar diet, insulin resistance, and developed along with aspects of metabolic syndrome, including hyperinsulinemia and obesity. It was found that changes in serum with diet and exercise that might be important include reduction in insulin, estradiol, insulin-like growth factor, and free testosterone with increase in SHBG and IGF-1 binding protein 1. Hyperinsulinemia stimulates liver production of IGF-1, plays a role in the promotion of prostate cancer, and it is the cornerstone for both metabolic syndrome and prostate cancer.
Bernard et al. Reported that after inducing insulin resistance with the HFS diet, the defect could be reversed by switching the person to a low-fat, complex carbohydrate diet before the person normalized their body weight, and in addition to controlling insulin resistance, switching to the LFCC diet also controlled the aspect of the syndrome. Metabolic syndrome and prostate cancer. During the investigation of relationship between prostate cancer and MS, Bernard et al. Suggested PC might also be an aspect of the MS. The hyperinsulinemia connected with the MS should decrease SHBG and increase the amount of free sex hormone available to connect with tissue receptors and enhance the hormone-related cancers, including PS.
Insulin resistance and hyperinsulinemia are the cornerstone of the metabolic syndrome, and as discussed earlier, can be induced by consuming an HFS diet. The HFS diet increases insulin receptor autophosphorylation, which results in insulin resistance and compensatory hyperinsulinemia. The exact cause of the decrease in receptor autophosphorylation is not known, but may be the result of the nitration of the tyrosine components of the insulin receptor, because they are readily attacked by the nitrogen radicals that are increased with an HFS diet. The body compensates for the insulin resistance by increasing the output of insulin in an attempt to avoid type 2 diabetes. The compensatory hyperinsulinemia of the MS may be the cornerstone of prostate cancer promotion. Insulin acts on the liver to reduce the production of SHBG and IGF-BP 1 and 2 while stimulating the production of IGF-1. These circulating factors are involved in stimulating proliferation and blocking apoptosis in prostate cancer cell lines. The changes in the IGF axis seem to be the most important because adding IGF-1 back to the serum after diet or exercise or both prevents the decrease in cell growth and blocks the decrease in apoptosis. Insulin also stimulates aromatase activity in fat tissue to increase estradiol production. In addition, N6 fatty acids can increase aromatase when the cyclooxygenase pathway is activated. Estradiol can stimulate the growth of prostate cancer cells, an action that might be achieved by interaction with the IGF-1 receptor. Studies from the Leawer laboratory shows that in cells with damaged DNA, IGF-1 could block the cells from going into apoptosis by stimulating the phosphodienosyl-3 kinase, which is also known as P13K, and mitogen-activated protein kinase, which is also known as MAPK pathway, resulting in degradation.
The information presented in this presentation indicates that prostate cancer is indeed another aspect of metabolic syndrome and is associated with consumption of a high-fat, refined sugar diet combined with a lack of regular exercise. Insulin resistance and hyperinsulinemia are the cornerstone of metabolic syndrome and are also factors for prostate cancers.
Insulin resistance and hyperinsulinemia precede other aspects of MS and may, in part, be an underlying factor in their development. Insulin can directly stimulate prostate cancer cell growth, but probably is more important as a result of its effect on the liver to decrease SHBG and IGFBP1, and to while increasing the production of IGF.
The changes in the IGF axis stimulate prostate cancer cell growth and more importantly inhibit apoptosis. Adopting a low fat, which is about 10 to 15% of energy with a balanced ratio of N6 to N3 fatty acids, complex carbohydrate diet along with daily 60-minute aerobic exercise will control MS in most cases and should reduce the risk of prostate cancer.
Thank you for listening to our presentation.